Okay, so I want to go over this paper called Galectin-3 and Galectin-1 bind distinct cell surface glycoprotein receptors to induce T cell death. So one of the reasons why I wanted to go over this is if you've seen my previous papers, I do believe strongly that Galectin-3 is able to work with Galectin-3 binding protein in order to help put you into this pro-inflammatory and pro-thrombotic or pro-blood clotting response. And the reason why I believe that is all happening is that there's a lot of labile, free labile iron that is being liberated and put into the body, which is driving all of those factors together. I should say it is tying all of these disparate symptoms together. So what I want to do is go over this so that we have a little bit more of an understanding of what Galectin-3 actually does, because Galectin-3 in of itself seems to have a lot of disparate or seemingly paradoxical effects on it. So with that, I will continue, and then I also later today will have a very informative video on how transferrin is also very important, or I shouldn't say transferrin, but how iron is very important in the regulation of certain hypoxia elements. So with that, let's continue. So galectins are a family of mammalian beta-galactoside binding proteins. So what is a beta-galactoside? So galactose is, if you have drank milk before, galactose is one of the subunits of the lactose molecule. So lactose will have glucose and galactose, glucose and galactose bound together, and you make this lactose disaccharide. And so, but for a beta-galactoside, just take the galactose and then have that galactose form a beta bond with a protein or some other biomolecule. Okay, so that is just what a beta-galactoside is. So galectins are a family of mammalian beta-galactoside binding proteins. So the galectins are proteins which bind to beta-galactoside, which could be on other proteins. So this will be a way for the galectins to bind to another protein. So and this binding positively and negatively regulates T cell death. So the extracellular galectin-1, and let's get an image of galectin-1 just so that we can see it, so that you can have an image in your head. So this is galectin-1, or LGAL-S1 would be the other name for it. So this binds to galectin, or I should say it binds to galactose, beta-galactosides, predominantly. So a lot of these lectins actually have a little bit of promiscuity as far as what carbohydrates they can bind to, but this one will preferentially bind beta-galactosides. So this galectin-1 will directly induce, or the extracellular galectin-1, so outside of the cell, when galectin-1 is outside of the cell, it induces the death of T cells and thymocytes. So the thymocytes are the immune cells that have migrated to the thymus in order to mature into T cells. So the thymocytes, these are, well, lymphocytes, so they are white blood cells that are associated with the with the lymphatic system. And so here, thymocyte, immune cells present in the thymus before it undergoes transformation into a T cell. So T cell, the T comes from thymus. So these thymocytes go to the thymus in order to turn into T cells. So this galectin-1, when it's from the outside of the cell, it can bind to the T cells and the thymocytes and cause them to kill themselves. So this would be a way to control or suppress the your T cell populations. Okay. So, however, the intracellular, within the cell, the intracellular galectin-3 will block the T cell death. So galectin-1 on the outside of the cell will cause T cell death, but galectin-3 on the inside of the cell will suppress or protect the T cells from death. So galectin-3 on the inside will protect your cells. So in contrast to the anti-apoptotic, which anti-apoptotic means anti-programmed cell death, the anti-apoptotic function of the intracellular galectin-3, we demonstrate that the extracellular galectin-3 directly induces the death of human thymocytes and T cells. So extracellular galectin-3 on the outside of the cell will have the opposite effect of galectin-3 on the inside of the cell. And so this is with specifically T cells, but there might possibly be other similar effects that are happening in other cells in the body that share the receptors as the T cells. So let's continue. However, events in galectin-3 and galectin-1 induced cell death differ in a number of ways. So the thymocyte subsets demonstrate different susceptibility to the two galectins. Okay, so there are subsets there are differentiated thymocytes that can fall into different subsets, and they have different susceptibility to these two galectins. So galectin-1, extracellular galectin-1, will kill double negative and double positive human thymocytes. So what does double negative and double positive mean? Well, this is in reference to CD4 and CD8. So let us look up, I'll just look up double positive human thymocytes and get the Wikipedia article up. So thymocytes. So the stages of maturation. So thymocytes are classified into a number of distinct maturational stages based on the expression of cell surface markers or cell surface receptors as well. The earliest thymocyte stage is the double negative stage, in which they are negative for both CD4 and CD8. So, and I'll go over what these are a little bit more later. So, which more recently has been better described as the lineage negative, which can be divided into 4 substages. So, and here are the double negative stages, and it's divided into 4 stages. So the next major step is the double positive stage, positive for both CD4 and CD8. Oh, sorry, yeah. And the final stage in the maturation is the single positive stage. So what happens when these proteins, or I should say when these T cells, these thymocytes, are maturing into their final form, they start off as double negative, and then they mature into double positive, and then when they are fully mature, they are then single positive. So they have become either just CD4 positive or just CD8 positive. So that is referred as the single negative state, and that is a part of the maturation process of these T cells from thymocytes. So what I will want to do is go over what is CD4 and what is CD8. So CD4, this is the cluster of differentiation 4, and it is a glycoprotein found on the surface of immune cells such as the T helper cells, the monocytes, macrophages, and dendritic cells. And it was discovered in the late 1970s. So let's see what it does explicitly. So CD4 is a co-receptor of the T cell receptor, and it assists the T cell receptor in communicating with antigen-presenting cells. So these are cells that are presenting antigens on their surfaces. So they are cells that have... Sorry about that, I had to rescue someone with a flat tire. So these antigen-presenting cells are going to be taking in antigens or consuming some of the things that are around them. And then they will process these proteins that they have consumed and then break them up into smaller peptides. And then they will take these small peptides and present them on their surfaces as antigens. So that is what the CD4 is working with the T cell receptors, so the receptors that are on the T cells, to interact with these antigen presenting cells. Okay, so the T cell receptor complex and the CD4 bind to the distinct regions of the antigen presenting major histocompatibility class 2 molecule. So this is the molecule, the MHC molecules are the molecules that have these small peptide fragments that are added to them, and those small peptide fragments came from the outside environment in some way. So, and I think that's all I really need to say about the CD4, except I should say that they do end up leading to the activation of transcription factors. So these are factors or proteins that influence DNA transcription into messenger RNA. So some of these include the nuclear factor kappa B, which I had done a video or a series of videos on previously. So that is the CD4 molecule. There is also the CD8 molecule, which it's cluster of differentiation 8. It is a transmembrane glycoprotein, so it also goes through the membrane of the cell, and it serves as a co-receptor of the T cell receptor. So both the CD4 and the CD8, those are co-receptors that will interact with the T cell receptor, or the TCRs. And so along with the TCR, the CD8 co-receptor plays a role in T cell signaling and is aiding the cytotoxic, or the cell-killing effect of the aiding with the cytotoxic T cell antigen interactions. So the CD8 is going to help the T cell receptor find these antigens and then kill the cells that have these antigens that are on them that are associated with pathogens or associated with apoptotic events. So sometimes cells want themselves to die. So I don't know if this is going to be specific. Okay, so this is like the T cell receptor, the CD8 will bind to the major histocompatibility complex molecule, but it is specific for the MHC class 1 protein. So remember, the CD4 cell was more specific to the major histocompatibility complex 2. This one is more specific to the major histocompatibility complex class 1 molecule. So, and just know that they will have slightly different, or they will have different I should say, cellular signaling pathways associated with them. So with that is the CD4 and the CD8, and then as the thymocytes, the cells that have migrated into the thymus, as they mature into the mature T cells, they start off as double negative, then they become double positive, and that they'll have both of them, and then as they finally mature, they will have either CD4 or CD8 expressing. So, and then they'll be single positive. So, going back to our original paper, the thymocyte subsets demonstrate different susceptibility to the two galectins. So the galectin ones will kill the double negatives and the double positive human thymocytes. So those are all the immature thymocytes, and they kill them with equal efficiency, so they will both die equally with galectin-1. However, galectin-3 will preferentially kill the double negative thymocytes, so these are the more immature ones. So the galectin-3 binds to a complement of the T cell surface glycoprotein receptors, distinct from those recognized by galectin-1. So this is saying here that the receptors that galectin-3 binds to are different from the ones that galectin-1 binds to. And this is important because this is something that can cause the differential outcomes of galectin-1 versus galectin-3. So remember, galectin-1 is killing both the double negative and the double positive with equal efficiency, which means they are probably interacting with receptors that are found on both the double negative and the double positive thymus cells. However, galectin-3 binds to a complement of the T cell surface glycoproteins that are distinct or different, which means those receptors are probably differentially expressed in the double negative and the double positive. I mean, this would be just one possible mechanism to help cause this differential apoptosis. So continuing, of these glycoprotein receptors, the CD45 and the CD71 are the ones that galectin-3 is interacting with to cause T cell death. Now, the CD45, let's look up what that is first, and this is Ptprc. It's a protein tyrosine phosphatase receptor type C, and just know it's an enzyme, and that it is going to take phosphate groups and add them to tyrosines of different proteins, and this is one of the ways that they can do some cellular signaling. So let's see, it is a transmembrane protein, and that is present in various isoforms. Isoforms meaning different types based on different alternative splicing of the messenger RNA, but it's found in a lot of different cells, and it's found on all differentiated hematopoietic cells. So these are all of the, except for the erythrocyte and the plasma cells. So basically, these are on all of the white blood cells, pretty much. So, and it functions through either direct or indirect, functions through either direct interactions with components of the antigen receptor complex through its extracellular domain, or by activating various SRC family kinases required for the antigen receptor signaling. Don't worry too much about this. Just know that there are many alternatively spliced transcripts of it, which might be one of the reasons why the double negative and the double positive white blood cells or thymus cells, thymocytes, that might be one of the reasons why galectin-3 might not bind to the same, to this CD45. Wait, is it 45? Yeah, it might not bind to the CD45 of those different types with equal efficiency, possibly due to the different isoforms or the different alternative splicing that is taking place. Don't worry too much about that. Let's see if... Yeah, I won't go into too much detail. If you want, you can go to this Wikipedia article yourself. But the one that I really want you to pay attention to is the CD71. So what is CD71? This is the transferrin receptor 1. So this is the receptor on the cells that are important in interacting with transferrin. So remember, transferrin is the protein that will take iron 3 plus, and move it throughout the body so that you basically can have iron homeostasis. It protects your body from iron while still allowing that iron to flow through your blood to get to the cells that need it. So the transferrin receptor 1 is a protein in humans encoded by this gene, and it is required for iron import from transferrin into the cells by endocytosis. So transferrin will interact with the transferrin receptor 1, and it will encourage endocytosis, which would cause the iron to be brought into the cell. So here we can see, here is a diagram of what we're dealing with. So here we have our transferrin over here, right, TF. This is our transferrin that is floating on the outside of the cell, that it has these irons that are bound to it, right? This transferrin receptor will take this transferrin and then cause endocytosis. So this would be an endosome after this endocytosis has happened. And here you can still see that original transferrin receptor with the transferrin still bound to it, but now you have these, the iron has become liberated, and so the iron is now existing in the endosome, and it gets turned into a labile iron and then gets pumped out of that endosome into the rest of the cytoplasm. So here we have the iron that's in the cytoplasm. Now, I'm not going to focus too much on this. Just know that this iron that is existing in the cytoplasm is going to have a lot of effects on the rest of the body. So one of the effects that we are going to see of this iron being put into the cell is that it is going to interact with these IRP12s, and also it's going to interact with ferritin, and by that it is going to, actually don't worry about that. Just worry about these IRP12s. And what are the IRP12s? Well, these are called iron responsive element binding proteins. Okay, so I think I'm going to do a different video on these iron responsive element binding proteins later today. Just know that there is a connection between the iron that is in the body and these and the activation or inactivation of these iron responsive element binding proteins. And so these binding proteins are going to interact with the mRNA and influence their translation rates. So your body has synthesized these from your DNA. It has synthesized messenger RNA, which can encode for a lot of proteins that are involved in iron homeostasis. And so as these iron responsive element binding proteins interact with their iron, then it prevents them from being able to bind to the mRNA. And this binding for the mRNA can either be downregulating, which happens with, actually I'll go into more detail in those other videos. Just know that this iron responsive element binding protein is influencing translation rates. And by inhibiting this translation rate, you are then creating a different protein profile or a different translation profile into proteins of the cell. So this is one of the, just know that this is one way to connect those two seemingly disparate systems. So, so just know that galectin-3 binds to CD71, and this causes cell death of the T cells. So in addition, CD7 that is required for galectin-1 induced death is not required for death triggered by galectin-3. So just know that CD7, which is one of the binding protein, or sorry, one of the receptors that is found on the cells that galectin-1 will kill, even if you get rid of that CD7, galectin-3 is still able to kill those cells. So this is saying that it's undergoing a completely different strategy of killing the cell. Anyway, CD45 remains uniformly distributed on the cell surface, in contrast to the CD45 clustering induced by galectin-1. Thus, extracellular galectin-3 and galectin-1 induce cell death of T cells through distinct cell surface events. So the cell surface events that galectin-3 and galectin-1 do to the cell surface of the T cells, those are completely different, which ends up resulting in the apoptosis. So however, as galectin-3 and galectin-1 cell death are neither additive nor synergistic, the two death pathways may converge inside of the cell. So what they're saying here is whatever downstream signaling that is taking place, if you add both galectin-3 and galectin-1 on the surface of these cells, somehow the signaling probably crosses into the same signal, because there is no additive or synergistic effect of doing both at the same time. So this would indicate that even though the cell surface responses are completely different, the internalizing of that cellular signaling converges somewhere, which can result in apoptosis. So with that, I'm going to just reiterate that galectin-3 is fulfilling an absolutely completely different role when it's expressed on the surface of the cells versus when it's within the cell. So those are two completely different responses. Now, what is something that I believe could initiate this? Well, it could be possible that galectin-3, while it's in the cell, let's say the cell explodes, right? Or you have some cell death nearby, that could theoretically release some of the galectin-3 that is within that cell from the inside of the cell to the extracellular matrix, right? And once this galectin-3 is floating around in the extracellular matrix, it then might be able to start binding with different cells, and then convince those cells that another cell died. Now, what I am imagining right now is, let's say you have a group of T cells in an environment, and they all have this galectin-3 inside of their cells, which is helping them prevent themselves from killing themselves, right? If there is enough galectin-3 in the extracellular matrix, and then it kills one of the T cells, right? So it binds to the surface of the T cell and then causes that T cell to die. What's going to happen is the galectin-3 that was inside of that T cell, as the T cell kills itself, it then releases that galectin-3 into the environment. And that release of galectin-3 will then bind to other T cells, and then and maybe it'll push them over the threshold for them to then say, oh, I should kill myself. So this could have basically, what would the word be? It could be a positive feedback loop that ends up killing a bunch of the T cells within the body. Now, granted, this might be, or this very well could be, actually this is definitely a highly regulated, important process right? So you don't always want to have your T cells be activated and living. Sometimes you want to suppress your T cell response for some reason. Perhaps after the disease has come and gone, okay, well, we no longer need these T cells, so start killing them because they're no longer useful right now. So that could be something that could be happening. Sorry, stuttering. So with that, I think I'm going to stop this video. And then my next video, I am going to go over a little bit more on what these iron responsive element protein 1,2s are and how this could theoretically be bridging between galectin-3 binding protein and galectin-3 and how do I phrase that? How some of the problems that we are experiencing with COVID-19 could be the result of the dysregulation of iron responsive element binding protein, which could be the result of having way too much labile iron in the solution. So if we have way too much iron 3 in solution, that theoretically could cause, how to phrase that? If we have a lot more iron 3 and iron floating around in our blood serum, it can get into the cell and then cause dysregulation. Well, first it'll be normal regulation of the iron responsive protein, right? And then we will have adequate changes in our protein synthesis to respond to that extra iron. But if we have too much, that could theoretically completely override the ability of these iron responsive proteins to function correctly. And I do believe that one of the negative effects of this might be hypoxia. So with that, I'm going to head on out. Thanks for listening, and hopefully I'll have this other video out by the end of the day.